All right, let's get right into it. We're going to talk about the story of the gene. And look, this is one of the most powerful, personal, and yeah, sometimes dangerous ideas in the entire history of science. It's not some dusty old story in a lab. It's a story that lives inside every single one of us, shaping our families and our futures. And the poet Philip Larkin, wow, he just cuts right to the heart of it, doesn't he? Heredity isn't some abstract thing you read about in a textbook. It's raw, it's intimate, it's that messy, complicated inheritance we get from our families. This verse, it really sets the stage perfectly for a journey that's just as much about family as it is about science.
So, starting with that poetic truth, let's turn to a real family. Because you see, the quest to understand the gene, it so often begins not with a microscope, but with a really personal, sometimes haunting question.
For the author Siddhartha Mukherjee, this wasn't some academic puzzle. I mean, this was the real story of his uncles Rajesh and Jagu and his cousin Moni. All of them brilliant, but each one consumed by mental illness. It was this pattern, this ghost haunting the family tree. And that personal mystery, that question of, what is this invisible force being passed down through my family? That's really our starting point for understanding the gene itself.
And that very personal question, well, it sends us hurtling back in time to join this centuries-long search for the secret code of life, the mechanism of heredity.
Man, the search for this thing was long and winding. You have to go way back to ancient Greece, where Aristotle had this unbelievable insight. He figured hereditary was about transmitting information, a message becoming a physical thing. Then, fast forward like 2,000 years, Darwin brilliantly explains evolution, but there's this huge hole in his theory. He has no idea how traits are actually passed on. And then you get Gregor Mendel, this monk just working in his garden who figures the whole thing out in 1865. But his work was so ahead of its time, nobody got it. It was completely ignored for decades, until boom, 1900. It's rediscovered, and that's what kicks off the entire field we now call genetics.
And the conceptual leap he made, it was just enormous. See, before Mendel, everyone just kind of assumed this idea of blending inheritance. You know, like mixing paint. A tall person and a short person have a child, they'll be medium height. Traits just mix and get diluted. But Mendel showed that's totally wrong. Traits are passed down in these discrete little packets, what we now call genes. They don't blend. A trait can be hidden for a generation, but it's still there, totally intact, ready to reappear. This idea, particulate inheritance, is the foundation for everything that came next.
Okay, so as we jump into the 20th century, the pace just accelerates like crazy. The gene is finally seen, it's understood, and it leads to one of science's greatest triumphs. But this new knowledge is also twisted into one of humanity's absolute darkest chapters. This was it, the breakthrough. 1953, for the first time we could actually see the code. Watson and Crick's double helix wasn't just some pretty shape, its structure immediately told you how it worked. You could see how this amazing molecule could store vast amounts of information in its sequence of letters, A, T, C, and G, and you could see how it could copy itself. Humanity could finally read its own instruction manual. Just think about that.
But at the exact same time all this amazing scientific understanding was growing, a really dark ideology was taking hold. The science of genetics was corrupted into eugenics. And it started with this seemingly noble goal, improving the human race, but it quickly spiraled into a terrifying social and political movement to control who was and was not allowed to have children.
And the logic of it just escalated with chilling speed. First, it was confinement, locking away people they called genetically unfit. Then, that progressed to forced sterilization. We're talking about laws that led to thousands of operations on people who had absolutely no say in the matter. And this dark path, it reached its absolute macabre peak in Nazi Germany, with the state-sponsored murder of people deemed lives unworthy of living. It's just a haunting reminder of how easily scientific ideas can be twisted to serve the most horrifying ends.
So, coming out of that incredibly dark history, we move into the modern era. And this is where the terrible lessons of the past collide with this awesome new power to manipulate our genes directly, changing medicine and our very sense of self forever.
So here's the basic toolkit of genetic engineering, and it's actually remarkably elegant. Scientists found these things like molecular scissors that could cut DNA at really precise spots. Then they figured out how to paste in a new piece of DNA, say the gene for human insulin. You stick that new recombined DNA back into something that grows fast, like bacteria, and suddenly those bacteria become tiny factories, just churning out life-saving medicines. This wasn't just theory anymore. This was the birth of the biotech revolution.
And with the ability to write genes came the ability to read them with stunning accuracy.
So what's this number, 70, 80%? Well, that's the estimated lifetime risk of getting breast cancer for a woman who inherits a specific mutation in a gene called BRCA1. And just like that, your genetic code could start telling you about your future.
This kind of knowledge literally created a new kind of person, the previvor. You're not sick, but you carry this profound risk that's written into your cells. Your whole relationship with your body, with your future, it's fundamentally changed. The question is no longer if you might get sick, but when. And what on earth are you supposed to do about it? That's a heavy, heavy burden of knowledge.
So here we are. We've learned to read the code, and we've even learned to rewrite parts of it. And this brings us right to this very moment, and to some of the most profound questions we have ever faced about what it even means to be human.
The latest revolution is a technology you've probably heard of, called CRISPR. The best way to think about it is like a biological word processor for the genome. It lets scientists find a very specific sequence of DNA, a single genetic spelling mistake, and correct it with just unbelievable precision. This isn't just cutting and pasting whole genes anymore. This is genomic surgery. We're editing the very letters of life. And of course, this incredible power forces us to face some monumental questions. I mean, the potential to cure devastating genetic diseases like Huntington's or cystic fibrosis is right there, almost within our grasp. But where do we draw the line? Is fixing a disease the same thing as enhancing a trait? Should we be making changes that our children and their children and all their descendants will inherit forever? And maybe the biggest question of all, who gets to decide what's a defect that needs fixing versus what's just a human difference?
The journey to understand the gene has brought us to this point. We're no longer just subject to our genetic code. We are becoming its authors. The power to direct our own evolution is, well, it's here. The story of the gene has become our own story. And the next chapter? That's one we have to write for ourselves.